Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speakers your questions. Your questions in the question and answer window will only be visible to myself and our speakers. So thank you for joining us today. It is now my pleasure to introduce our speakers, Aurélie Tacheny and Jorge Escobar from Eppendorf.
Hello everyone and welcome to today's webinar entitled Stem Cells Derived Exosome Production in Stirred Tank Bioreactors. This is the agenda we prepared for you today. We divided in three main topics. First, the introduction. We will detail the exosomes as a promising cell-free therapy tool, and then we will discuss about the challenge behind exosome production process. Later, we will discuss about process development in DashBox mini parallel bioreactor system, and finally, the exosome production on the Siberia twin bioreactor control system. Stem cell therapy, also known as regenerative medicine, is designed to repair damaged tissue in the human body by implanting new adult stem cells, modulating the immune system and reducing inflammation. There are 5,000 clinical trials registered for different biomedical applications so far. The premise of the cell therapy is to help the body to heal itself, increasing the quality of life for patients. The global stem cell therapy market is a very competitive market and is estimated to reach around $146 million by 2021. This market is fueled to the therapeutic action of the stem cells for several disease treatments and because of the stem cells line banking. In addition, induced pluripotent stem cells line has also contributed to the growth of this market due to the social ethical issues associated with embryonic stem cells. As you can see here in the slide, the musculoskeletal disorder segment dominate the global stem cell therapy. Unfortunately, clinical trials have made only a modest improvement in restoring normal functions to the degenerative tissues. In the earliest days, the mechanism of action of the cell therapy claimed that cells implanted should integrate in the site of injury, replace the damaged tissue, and improve tissue and organs functions. However, limitations associated with cell localization and cell survival in the target tissue works against tissue regeneration. But the real question here is, do we have the right tools to address the cell therapy limitation? Let's explain a little bit more about that. Exosomes were discovered more than 3 years ago and were considered cellular waste disposal. According to this mechanism represented in this picture, exosomes are one of the most important paracrine effectors involved in intercellular communication. Think of an exosome as an envelope with instructions inside, like one cell mailing a letter to another cell and telling it what to do. In this case, the envelope contains microRNA, non-coding RNA, or proteins. In addition, the exosomes are released from many cell types, including T and B cells, mesenchymal stem cells, tumor cells, etc. They mediate an intercellular or distant communication between cells, and also they modulate immunoregulatory processes, set up tumor escape mechanisms, and mediate regenerative processes. Also, they have round-shaped morphology, and they are present in the majority of body fluids such as blood, serum fluid, urine, amniotic fluid, etc. The cell secretome consists of extracellular vesicles, specifically exosome, microvesicles, membrane particles, peptide, small proteins such as cytokines, etc. In this case, exosomes are lipid bilayer vesicles with a diameter ranging from 50 to 100 nanometers, which can be easily confirmed by surface markers such as CD9, CD63, CD81 of the transplants. Of course, not all the mesenchymal stem cell derived exosomes are similar. They are different. They are coming from different sources, and they are present in various adult tissue. Also, exosome yields and content could vary. So they offer several advantages compared to cell-based approaches, including exceptional stability and biocompatibility. Moreover, they can be delivered to the target tissue easier than whole cells and can migrate across the blood-brain barrier. According to PubMed, the number of citations has grown more than 10 times in the last decade. Today, this new cell-free therapy has had a great impact in various fields of application, reaching around 192 studies or clinical trials worldwide related to exosomes that are already listed in clinicaltrials.gov. They have been used as drug delivery systems, as tumor biomarkers, and they have great potential also in bone and cartilage regeneration. As you can see here in this picture, there are important landmarks that I would like to share with you related to biotherapeutic particles story, with the approval of the first cancer vaccine, the first gene therapy, and the first oncolytic biotherapy product. And now my colleague Aurélie will discuss the challenge behind exosome production processes.
Now you have a clear view of what is an exosome and their growing interest in the context of cell-free therapy. Nevertheless, exosome production process is still challenging. The preclinical and the clinical development of exosome technology as a cell-free therapy and drug delivery platforms require large quantity of exosomes. It's really difficult to define a precise range regarding the optimal therapeutic doses. Indeed, it depends on the type and the purpose of this therapy and every specific study, and on the way exosomes are quantified. But here you have an example of the huge quantity of exosomes required to be administered in a mouse to achieve biological outcomes. Mass production is then required to generate clinically relevant amount of vesicles. However, exosomes mass production is challenging for different reasons. A delicate equilibrium should be reached regarding quantity and quality. The final product must be pure and keep its biological properties. And to reach this, the manufacture process have to be scalable, be controlled and standardized in order to improve reproducibility, and finally be in accordance with regulation requirements. But most of the current methods used to enrich, isolate, and characterize exosomes are not fitting with those aspects. The exosome production workflow is a multi-step process and could be divided in three major steps consisting in the upstream process, during which the cells used as exosome sources are expanded and exosomes are enriched in the conditioned medium. The downstream process, consisting in the isolation of secreted exosomes from the other element collected in the conditioned media. And finally, the quality control based on exosome characterization. And all those steps could be a source of variability and of challenge for researcher. For example, regarding the upstream process, researcher could have some question such as which exosome source to select or how to ensure efficient cell proliferation. So before to go in more detail in the upstream process, which is the topic of today, I would like to describe briefly some challenging aspects and key message associated with the downstream process and the quality control. A large variety of techniques could be used to isolate exosomes. Historical gold standard methods are based on ultracentrifugation, with two main variations, either differential pelleting approach or using density gradient. But from a manufacturer perspective, ultracentrifugation-based methods are very difficult and present many limitations. It's difficult to scale up. The process is long with successive isolation steps and require high level of skills and expensive equipment. Other techniques exist, of course, to isolate exosomes, such as size exclusion chromatography, tangential flow filtration, or precipitation-based method or affinity capture method. Here are some key important messages to consider for transition from bench scale to clinical production of exosomes regarding isolation strategy. First of all, for the moment, there is no perfect solution. There is really a lack of standardization for exosomes isolation. However, as you can see on this picture, isolation strategy you will use will impact the final product. Combined techniques are often used in order to improve purity of the final product, and the strategy have to be really select depending on the final purpose. For example, in the case of therapeutic purpose, the crucial aspect to keep in mind for exosome mass production process are scalability, regulation requirement, potency maintenance, process control, specificity, and efficiency in terms of cost, yield, and time, of course. There is also a large variety of methods used to characterize exosomes, making result and analysis difficult to compare from a study to another one. Some methods are based on physical characteristics and properties, others on the presence of specific markers, and recent review also highlight emerging techniques. But once again, there is some challenge, such as standardization, sensitivity, specificity, and throughput. Now we will focus on the first step of this workflow, the upstream process, resulting in an efficient exosome secretion. We would like to highlight key factors to address in order to improve and warrant a successful and reproducible upstream process. During the first part of the introduction, we introduced the fact that not all exosomes are similar. Their content could be cell type specific, for example. But very importantly, cell culture parameters used during the ex vivo cell expansion will also largely impact quantity and quality of secreted exosomes. Any change in the cell phenotype due, for example, to adhesion, proliferation, or any cellular stress could cause variation in the composition and the function of the secreted exosomes. Culture medium could impact, of course, but also culture support and other physicochemical parameters used during the cell expansion process. Regarding the cell culture medium, in order to obtain a robust cell proliferation, culture media are usually supplemented with FBS as a source of growth factors. However, this kind of animal-derived supplements contain large amounts of exogenous exosomes, which could contaminate the vesicles of interest. In order to avoid the contamination, you can use different strategy. Either you can use exosome-depleted FBS to reduce the potential contamination, but you can also process in a two-step process with a first phase consisting in the cell expansion in a classical culture medium supporting cell growth, followed by a second step. As soon as the sufficient cell number is reached, a change of medium for a medium of collection is performed, and this medium is usually animal-derived component-free. Some commercial version exist for this kind of medium, and the composition of those medium are optimized in order to not push the cell proliferation, but push the exosome secretion. Of course, cell culture support will also greatly impact the support on which the cells are expanded. Conventional 2D adherent cell culture support, such as flask or multi-layer flask, present some limitation in terms of growth surface area, for example, per volumes. More physiological and also offering easier scalability, the 3D macrocarrier-based cell culture in a bioreactors is particularly interesting in this context of exosome production. And several teams have already demonstrated and published that the 3D culture is beneficial for exosomes yield and quality. 3D culture parameters should be optimized in order to improve attachment of the cells on the carrier, but also an optimal cell growth. And in terms of parameters, we can optimize the type of carrier, the initial cell seeding quantity, the cell microcarrier ratio, of course, and also all the first phase of initial adhesion of the cells on the carrier with or without agitation, for example. Regarding cell culture parameters, different parameters could impact. It could be the pH, the oxygen tension, of course, and we know that some lower oxygen tension could be closer than the in vivo environment of stem cell, for example, and could be beneficial. The temperature could also impact agitation or the type of feeding strategy you use. But very importantly, you have to optimize and maintain a stable and a more physiological environment as possible and avoid any cellular stress. In this context, and in order to ensure a stable and standardized upstream process, bioreactors present numerous advantages. They ensure continuous monitoring and control, ensuring maintenance of optimal culture parameters through the entire process. They also offer scalability with similar vessel geometry capabilities at different sizes. So it's really convenient in this process. To conclude this introduction, we would like to insist on the importance of the standardization through the entire process and the entire workflow, from the cell culture to the final product characterization. All details about protocols and strategy are really crucial to be published in order to allow comparison between studies. In the next part of the presentation, we will introduce you the use of the DASBox mini bioreactor system to develop an exosome production process in small-scale tire tank bioreactors and optimize culture condition before potential scaling up. Here is a summary of the experimental strategy used for the upstream process in this experimental work. Mesenchymal stem cells derived from adipose tissue have been selected as exosome sources. After thawing, cells recover a few days in a flask. The appropriate amount of cells is then seeded on microcarriers in a BioBlue single-use vessels. Cell growth is monitored during 5 days by the DASBox system, and growth factors have been had 3 days post-seeding in order to improve the cell growth. Regular monitoring of the cell adhesion and viability are performed during this period of expansion. After 5 days of cell growth, culture medium have been completely changed for a collection medium free of exogenous exosomes. Exosomes enrichment occurs during 2 additional days before harvesting and downstream processes. Here you have the main process parameter used for the upstream process. You can see, for example, the type of macrocarriers we have used, coated with Syntimax in order to improve the initial adhesion, the inoculation cell density, the working volumes, the ratio between cells and carriers, which is also very important, the dissolved oxygen percentage we have used, 40%, which means 8% pressure of oxygen, which mimic the in vivo environment, the pH, the temperature, the gas flow, and of course, parameters of agitation. As mentioned here, during the first hour, during the first contact of cells with microcarriers, we have stopped the agitation in order to improve the initial cell adhesion. After that, the agitation has been set at 100 RPM. Cell viability has been determined regularly during the process by staining the cells with a fluorescent molecule, the calcein AM, and visualized under fluorescent microscope. As you can see, through the 5 first day post-seeding, cells growth and progressively form small aggregates. Cell proliferation has been also evaluated through cell counting, performed at different time points during the cultivation. Samples of the cell suspension are taken from the bioreactors, and cell count is performed using a ViCell automated cell counter after trypsinization from the carrier, of course. Results obtained confirm the efficient proliferation with a very short lag phase here. Final cell density is noted here. And if we compare the initial and the final cell density, we have obtained a fold increase of 34.7. Metabolic profile of cells is also followed. So lactate and glucose concentration have been monitored at different time points during the process. The arrow here indicated the change of medium we have performed after 5 days for the collection medium. Here you have the experimental process used for the downstream part. After collection of the EV conditioned medium, the sample is centrifuged at low speed, filtrate, and concentrate. A small sample is taken from these volumes to perform the particle size distribution analysis. But the rest of the samples is completely engaged on a size exclusion chromatography column. We have collected 22 fractions, and the total protein content has been evaluated with a nanodrop. After the chromatography, every fraction have been concentrated and tested through 2 different methods, a specific ELISA test for CD63 and a specific Western blot for the same specific marker. You have to note that we have performed a negative control. This sample was run for each experimental condition and consists in the collection medium, so here, but not in contact with the cells. And this non-conditioned medium follow exactly the same process than the conditioned medium. Here you have the result obtained on elute fraction. In blue, the total protein amount of each fraction. On top of that, you have in red the specific expression of the CD63, specifically expressed in the membrane of the exosomes. As you can see, high relative content of CD63 was found in the fraction 16 to 20, with a peak in the fraction 19, corresponding to a relative quantity of 9.1 10 to 10 exosomes in this fraction. The negative control show analogous profile on total protein, but without any expression of the CD63. The Western blot result confirmed those results with the ELISA assay with a distribution of the CD63 protein in the same fraction, so from 16 to 20. Finally, a particle size distribution analysis has been performed with 2 different techniques, the DLS for dynamic light scattering and the CLS for centrifugal liquid sedimentation. As you can see on those 2 pictures, we obtain population of vesicles with expecting size comparison with exosomes. So with those results, we could say that the DASBox mini reactor system is a perfect tool to test and optimize the process at small scale. You can compare the influence of different external experimental parameters of culture in parallel. Every physicochemical parameters are monitored with in a very stable, defined, and reproducible environment. I will now let Jorge present another strategy to produce exosomes with another system, the Cyvario.
So thank you, Aurélie. So a different strategy was followed in this study. So we use BioBlue 1C single-use vessels as bioreactor for human-induced pluripotent stem cells, iPSC, derived mesenchymal stem cells, exosome production, isolation, and characterization. We employed the Cyberio-Twin bioreactor control system, as you can see here in the picture, as a controlled device, and collagen code microcarriers are cell culture support. So as you can see also, the main aim was the exosome production in 1 liter single-use vessel as a model for large-scale production in bioreactors. To demonstrate the ability of human iPSC-derived mesenchymal stem cells to secrete exosomes, we performed 2 medium exchange experiments in 4 steps, as you can see here in the picture. Step 1, detail the inoculum preparation. We use deep flask culture conditions for initial cell expansion and hyperflask in the third passage to achieve the appropriate inoculation cell density. Then, the stemness capacity of the iPSC-derived mesenchymal stem cells was analyzed by flow cytometry, noting that they display a typical phenotype of mesenchymal stem cells, positive for CD90 and CD29, typical mesenchymal stem cells markers, and negative for hematopoietic markers such as CD34 and monocyte markers like CD11b. Then the iPSC-derived mesenchymal stem cells were transferred into glass bottle containing collagen-coated microcarriers and cell culture medium. In this case, 2 different media were used, the MNF12 or ATCC complete media. So we kept the bottles in the bioreactor for 2 hours at 37 Celsius degree and 5% of CO2 under static conditions. On the other hand, prior to the preparation of the BioBlue 1C single-use vessel, we performed the calibration of the pH sensor and finally was sterilized. Then we outfitted each BioBlue 1C, among others, with a deep tube along with a compression probe adapter, both inserted in a spare PG 13.5 port under accepting conditions in the biosafety cabinet. The deep tube was used to do the medium exchange. Also, 3 liquid addition ports were used, one for the inoculation and medium addition, one for the addition of the base, and another for the addition of 0.1% of antifoam. In this case, we used pluronic F68. We then placed the assembled vessel in their respective temperature block controls to maintain the constant temperature. Finally, we introduced the MEN or ATCC complete media into each vessel and conditioned the vessel for at least 24 hours under the following parameters and set points. As you can see here in this slide, as I mentioned before, we performed 2 different experiments, and then the majority of the parameters are the same, but with certain difference, such as the inoculation density and the cell culture medium, pH, and overlay gas flow. In addition, as you can see in the right side, I would like to share with you a visualization of the DO cascade parameters used in this experiment. Following incubation, the 300 milliliters of cells and microcarriers were then transferred to the BioBlue 1C bioreactor, already filled with 700 milliliters of medium. After 5 days of cell culture, we perform a medium exchange every 2 days until day 9, and then on daily basis as required. But how we did that? So we stopped the agitations and gases flow for 5 minutes, allowing the microcarriers to settle at the bottom of a BioBlue 1C and exchange 10% of medium. using the deep tube to remove the cell culture medium from the surface and using the feeding pore to add fresh medium. After day 5, 8, 11, and 14 days of culture, we collected 50 milliliters of iPSC-derived mesenchymal stem cells along with microcarriers and medium using a container bag with line set. So we transfer to 50 milliliter, these 50 milliliters into 50 milliliter conical tube, allowing the iPSC to settle in the bottom of the tube. We removed the supernatant and added an additional 50 milliliter of medium using the same composition, but only changing the FBS, but FBS with 2% by 2% of exosome depleted FBS. Finally, we incubated iPS-derived mesenchymal stem cells along with the microcarriers for 48 hours at 37 Celsius degree and with using 5% of CO2 and 50 RPM. Then, approximately 48 hours after each collection day, we remove the supernatant and perform the isolation and purification of exosomes according, as you can see here, with ExoQuit TC Plus kit protocol with some modifications. So we collected a sample every day in the course of the first experiment, or every 2 days in the second experiment from the bioreactor to determine the cell viability, the cell density, and metabolite concentrations such as glucose, ammonia, and lactate. Let me explain a little bit how we did that. So 3 milliliters were collected for analysis. So we transferred that 3 milliliters in a conical tube, and then we washed the microcarriers with the PBS. Then later on, we added 3 seams in order to detach the cells from the microcarriers and filter the whole system through a 40 microns strainer. And the cell viability and cell density were analyzed. As you can see here in the picture, after the initial cell expansion, the cell count was erratic, especially after each collection day. So we attribute that or this behavior to a low inoculation density and the use of DMNF12 cell culture medium, whose composition may be suboptimal for the iPSC-derived mesenchymal stem cell expansion. But I have to say that overall, the cell growth increased fourfold more than the initial cell density on day 15 of culture, but the final density was low. So as I mentioned before, we performed a second experiment to increase the iPSC-derived mesenchymal stem cells inoculation density and the exosome production yield for each samples. In order to achieve that, we changed the cell culture conditions. So first, we perform the cell expansion and the initial cell attachment to the microcarriers in a TCC complete medium and increased the inoculation cell density. And finally, we kept the rest of the conditions the same as for the first experiment. As you can see here in the picture A, we observe an initial lag phase 24 hours after the inoculation, followed by the steady increase of cell growth between day 1 and day 9. The culture reached the stationary phase around day 9, followed by a subsequent decrease in cell density until day 15. Also, we have observed a substantial microcarrier aggregation late in the course of the run. Therefore, base of cell count in the later stage on only the floating microcarriers, thus the true average of this cells expansion in the vessel it should be much higher. In addition, as you can see in picture B, we determine the consumption of glucose and production of lactate and ammonia during the whole run. So, as shown in the picture, exosomes are highly rich in proteins in which the transponins membrane that are membrane proteins such as CD63, CD9, and CD81 and CD82, as you can see here in the surface of the exosomes, play really a key role in the cell's invasion, penetration, and function and fusion events. We use an ELISA assay, specifically ExoELISA Ultra CD63 kit, to quantify the exosome abundance. So we incubated the exosome with a primary anti-CD63 antibody that binds the transponin protein CD63 on the exosomal surface. The results show that the number of iPSC-derived mesenchyma stem cells exosomes constantly increased from day 5 to day 16. In addition, so we found also a direct correlation between cell density and secrete exosome up to day 9. So finally, to study the three-dimensional cell morphology on collagen-coated microcarriers, we collect samples at day 5 and 9, as you can see here in the pictures, and visualize them through bright field images. We evaluate the morphology, demonstrating that at the early stage, the cells in the cell growth profile, only a few cells, as you can see, are attached to the microcarrier surface, and progressively they form intermicrocarrier cellular bridges and aggregates, as you can see in picture B. It is well known that the 3D cellular environment allows the cells to mimic the in vivo cellular behavior involving cell-cells and cell extracellular matrix interaction and promoting cell signaling and proliferation. It's well known that recently have been reported that although the mechanism of exosome biogenesis, as I explained before, is not still well understood, findings suggest that exosome secretion and molecular cargoes can be altered by the cell's microenvironment. Finally, I would like to say that these Siberia twin experiments are preliminary studies and have not yet been optimized to obtain a maximum exosome production level. However, our observations can serve as a guideline for further improvement in mesenchymal stem cell-derived exosome isolation, purification, and scale-up protocols. Thank you.
Okay, great. Thanks. So the first question is, what type of microcarriers have you found increases MSC expansion the most? Is a matrix necessary?
Yes, perhaps I can answer to this question. In fact, that's right, that it exists a large variety of different carriers in the market. So some of them present surface coating to improve the cell adhesion. So how to select the best carrier for your cells? In fact, it will, of course, depend on the cell type you will use. For MSCs, for example, very good results have been published with various type of carriers like Cytodex3 or Cytodex1. But importantly, it will also depend on the composition of the culture medium. For example, synthetic medium or medium which are poor in FBS could require some help to improve the initial cell adhesion. In the case of exosomes isolation, you do not have to recover the cells at the end of the process. So you can really help the initial cell adhesion with the coating without risk of difficulties to recover the cells, because you don't need the cells at the end. And it's reduced the risk of initial lag phase. And in our work, we have used macrocarriers coated, in the case of Jorge, with collagen, and in my case, with Synthemax, which is a synthetic coating with RGD motif to help the cells to adhere and to mimic more closely the in vivo extracellular matrix environment. So it could really help the cells to adhere.
Okay. And how do you ensure that you do not have exosomes from a growth media in your experiment?
I can answer these questions. Yeah, you have to avoid bovine exosome contamination from the FBS. In order to do that, you can use your regular FBS and then also the high-speed centrifugation method to deplete the exosomes in the FBS before you culture, or you can use also the exosome deplete FBS from the market.
And the content of exosomes may be variable. How can we produce exosomes that contain consistent content, for instance, that are suitable for COVID-19 or other types of diseases?
I would say that the subpopulation of the extravesicles you will isolate, and of course their specific activity, will be very greatly impacted by all steps of your process, as we have explained in the introduction. So from cell culture parameters to the isolation strategy you will use. So to be sure to really limit the variability in the final product, I would say that standardization is really mandatory for every step of the process. So any change in the process could generate inconsistency in the content of your exosomes. And for this reason, it's really important to keep this aspect in mind from early R&D to large-scale exosome manufacture process. For example, by directly working with a scalable culture system, such as, in this case, the 3D culture on microcarriers with a bioreactor.
At day 5, did the microcarriers form aggregates?
Yeah, I can answer these questions. I mean, in my experiments, we detect the aggregate formation starting from day 9.
Okay. And as known extracellular vesicles from 2D culture had been tested in many different assays showing their biological effects, what about extracellular vessels from the bioreactor, or vesicles?
Yeah. Of course, the goal of the present work was not to characterize in detail the biological activity of the purified vesicles, but more to illustrate the compatibility of the Eppendorf solution in this field. But nevertheless, a number of recent publications describe the positive impact of the 3D culture on the exosomes quantity and also the exosome quality. And some paper, for example, describe that 3D culture-derived exosomes are closer to in vivo circulating exosomes, showing, for example, that the content or the small RNA profile of those exosomes are very similar to the in vivo circulating exosomes derived from plasma of patients, for example. So we are convinced that 3D culture could be very beneficial.
And too much cell microcarrier aggregation may suppose a drawback on exosome production?
I'm going to answer this. I mean, this is a good question. It could be, but we have to remember that this is a 3D cell culture environment, and the microcarriers that we use have a large surface area. So, and also that the exosomes are in the nano size range, so they can easily diffuse into the medium. But we didn't study how that parameter influences cell aggregation.
Oh, sorry, I was on mute. What is the input and output amount of the DASBox of the DASBox mini parallel bioreactor system?
That's for me. In terms of input for cells, we have used an initial quantity of 10 exponential 6 cells, and the output after 5 days of culture was of 10 exponential 8 cells.
But in terms of particles, if we pool the different fractions collected containing exosomes, we could estimate we have isolated exponential 11 particles, in total.
Okay. So it looks like we have time for just one more question. Thank you for all the great questions. If we didn't get to your question, we will be passing those directly to Aurélie and Jorge, and they can follow up with you directly. So the last question for the presentation is, have you conducted experiments to determine the combined effect of large-scale expansion of MSCs and bioreactors along with serum deprivation, and does this negatively affect the cells and subsequently the extracellular vesicles?
I will answer this question. This is an interesting question. So in my case, we performed several preliminary studies to carry out the cell culture in culture medium without FPS, and I can tell you the cell growth was severe affected. As you saw in our exosome production strategy, so we decided to culture the APS-C in a complete medium, so medium with the regular FPS. And then on every collection day, 40 milliliters of medium were exchanged with medium supplemented with exosome depleted FBS. I can tell you this is one of the biggest challenges in the field right now.
Okay, great. Well, thanks, both of you.
Thank you.
Thank you.
And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Goodbye.
Bye. Goodbye.